Yeah. Good afternoon. Sharon, is my screen visible now in the full view
Yes.
screen mode? Yeah.
Yes.
Okay. The first experiment is determination of pH. Okay. So very first experiment we will be starting with the pH determination. Okay. Yeah. The pH probably would have come across. Refers to the measure of hydrogen ion concentration in a solution. Please make a note down. So these are the points are important for your MCQs. Okay. The term pH refers to the measure of a hydrogen ion concentration in a solution. And the exact definition of pH is the negative logarithm of hydrogen concentration in water or wastewater. So please make a note on negative logarithm of hydrogen ion concentration in water. If I say example 10 power minus 7 like of minus 7 of say example the water, what is the pH? You can immediately say that is 7. Okay. So normally the pH ranges from 0 to 14. Okay. There is no zero value, but it normally may say that pH values is 0 to 14. Less than 7, you can call it as is the acidic range, and greater than 7, you can call it as is the basic range. That means 0 to 7, you can term as acidic, and 7 to 14, you can term as basic value, or base or basic. Okay. And if the concentration of hydrogen ion as well as the hydroxyl ions are equal in nature, that is termed to be neutral pH. Please make a note of that. So 3 points we discussed. And hydrogen ion concentration in a solution, you can refer it as pH, and you can calculate the pH or find out the pH of the solutions by taking negative logarithm of hydrogen ion concentration in the particular solution. And less than 7, that means 0 to 7, you can call it as acidic range, and 7 to 14, that you can say base or basic range. And if the concentrations of H plus as well as OH minus ions are equal, then it is termed to be neutral. Say example, drinking water, probably you may come across, it's a neutral pH. That means it's exactly 7 or 7.1 or something, 7 value. Okay. So you can have a look on the pH range normally, what you have come across. Okay, you can start from 1 to 14. Okay. So in our stomach, gastric acid may have a acid pH value of 1, and lemon juice a pH value of 2, apple juice is about 3, a tomato it is 4, black coffee it is about 5, and milk what you have it is about 6, and pure water it is 7. That's what I told you. Drinking water is neutral in pH, and egg, baking soda, and hand soap, ammonia solution, and bleaching powder, and concentrated solutions of any alkalis will come under base. So most of the food products what we eat is in the range of almost 1 to almost is 8.5. So please make a note on this, 1 to 8.5. Okay, and if you have a habit of using so much amount of like the juices like apple or lemon or tomato, obviously you may get a pH range of in your stomach in the range of almost 3 to 3.5. So that is also not good. People used to say that it is not good for your health. Sometimes it can damage your intestine or your inner pod or something. Okay, yeah. Why pH is important? That something is important. Why pH is important? So this is what you have to write in your experiment. So mere doing the experiment is not necessary, but by understanding the value or looking into the value, you are in a position to say some significance or what is the importance. Okay, so determination of pH is the one of the important objectives in the biological treatment of wastewater. Okay, please make a note. Probably you'll be studying now biological treatment of wastewater. I need not go into detail. In the case of anaerobic treatment, pH goes below 5 due to excess accumulation of acids, thereby your system or the process is severely affected. Please make it note down. If you have any difficulty in your stomach, normally any anaerobic system, normally people may consider your digestion system or your stomach. Okay, if anything on falls below 5 pH range, it will also create the accumulation of acids, and complete process will be affected. Probably you may say that system in medical field they may call it as ulcer. Okay, because of continuous accumulation of acids because of these conditions. And the pH, any changes from 5 to 10 in the case of aerobic treatment of wastewater, there could be a upset. Please make a note down. If it is less than 5 is also problem, and greater than 10 is also problem in the case of aerobic treatment. Okay, yes. And if it is any changes in like the pH, normally it is being adjusted by the addition of suitable acid or alkali. Probably would have come across if you have any something problem with they may use a lot of antacids. Probably would have come across. Okay, as the if it is his pH is very high, and you can add acid to neutralize. If the pH is very, say example, very high, you can add acids to neutralize, and if the pH is very low, you can add alkalis to neutralize the pH. Okay, please make a note on this. Okay, so thereby the pH value is very much important in the case of any chemical reaction, and the chemical shall be highly effective with a particular pH value. This also you would have studied in the last semester during coagulation. Coagulation normally it has been influenced greatly by the pH value. Consumption of chemical. If it is in a comfortable pH range, the consumption of chemical will be lesser, thereby the operational cost will be minimum. But if it is other way, you have to add excess chemical, which further reduces your operational cost. So please make a note on chemical coagulation. Last semester you have studied disinfection of water by bleaching powder or the chlorine chemicals, water softening by addition of lime and soda ash. Okay, so and corrosion control are governed by pH adjustment. Please make a note on. So what I mentioned already is importantly with aerobic treatment, important with anaerobic treatment, and we quantify the chemicals. You need to have an approximate range of chemicals, and a certain chemical are highly effective with a particular pH range. And for chemical coagulation, disinfection, water softening, and corrosion control are governed by pH adjustment. Okay, yeah. And for dewatering of sludges, probably you would have now, or you will be studying in this semester, dewatering of sludges or conditioning of your the chemical sludges, and further into dewatering, and you should have optimum pH range. Oxidation of cyanides, you should have the optimum pH value, and reduction of hexavalent chromium into trivalent chromium also need a favorable pH range. Please make a note on when CR6+, 6+ to CR3+, it will also go with a favorable pH condition. There could be a reduction of CR3+ to CR3+. Okay, yeah. And for any calculation, please make a note on any calculation of carbonate, bicarbonate, and carbon dioxide, corrosion, stability of water, that we call it as stability index. Everything depends on the pH value. So please make a note on any carbonic acid system is greatly influenced by the pH of the water. Okay, yes. And for your taste, please make a note on pH. If it is lesser than 4, will produce a sour taste. Higher than 8.5, it will give a bitter taste. Probably would have come across these difficulties in groundwater system. Okay, if you happen to take groundwater, and you may feel these situations 4 as well as greater than 8.5, sour taste and bitter taste. Okay, and any changes in the pH of water, sometimes it will also alter the scale formation. Please make a note on scale formation would have come across in pipes carrying with a high amount of calcium and magnesium, or the calcium hardness water. Okay, there could be a scale formation, or there could be a scale formations in your heating apparatus, in geysers or something. So there it is greatly influenced by pH value. And the last point is very important. That's what we have discussed in disinfection. Please make a note on. And if it is any organic matter present in the water during disinfection, there could be a formation of disinfection byproducts, DPP. Last semester we would have studied disinfection byproducts. Okay. The organic matter, as it is in the water, may not have any problem, but organic matter reacts with your applied chlorine, it will form a disinfection byproducts. One of the disinfection byproducts are trihalomethane, THM. Please make a note on THM. Okay, trihalomethane, and which are causing the cancer in human beings. That's what we would have studied in the last semester. Before disinfection, you should remove all the organic matter well in advance. Okay, so these are some of the important points you have to remember in the pH. Please, I'll repeat again. One is biological treatment. pH is important. In the anaerobic treatment or aerobic treatment, the pH ranges is very important. Any chemical calculations, you should also note down the pH range value. And the processes like coagulation, disinfection, water softening, and corrosion control are completely governed by pH adjustment. And the oxidation of cyanides in the case of industrial wastewater treatment, the reduction of chromium 6 into chromium 3 plus that need to have a favorable pH range. Any carbonic acid systems like carbonate, bicarbonate, and carbon dioxide system is greatly influenced by pH of the particular solution. And the taste with this is less than 4, it will give a sour taste, and greater than 8, it will have a bitter taste. And pH of the solution or water is greatly affected by scale formation as well as your heat scale formation in water heating apparatus, especially on geysers. And last one is about high pH value induces the formation of disinfection byproducts. So one of the example is trihalomethane. Yeah. Okay. So pH you would have already come across in the first year that pH principle, how to measure the pH. pH is being measured in terms of pH electrode in a combined glass electrode. Please make a note on this. Something is important. So pH electrode in a combined glass electrode. Combined glass electrode. Okay. So combined glass electrode consists of a sensing half cell, a reference cell together form an electrode system. Please make a note on the electrode system consists of sensing half cell and reference half cell. Please make a note on these 2 points are important. Okay. Yeah. The sensing half cell is a thin pH sensitive semi-permeable membrane, okay, and which is being separated by 2 different solutions. One is the outer solution, the sample to be analyzed, and you can also have an internal solution which is enclosed inside the glass membrane, has a known pH value. Please make a note on. I'm not going to discuss in detail in chemistry part. This and all you would have covered in your +2 or in the first year chemistry. Okay, so please remember how the working principle of pH meter. Okay, yeah, and you can measure the electrical potential because of this. The half cells, and there could be a electrical potential is developed inside and also another outside. Okay, the difference in potential is measured in terms of pH of the particular sample. So please make a note on this. Slide is important. How the pH is being measured in a particular solution. Okay, so these electrode types you would have come across in +2 or first year. You have what is this glass electrode and what is this your reference electrode, and whatever it is pH is being measured in a combination electrode. And you can also measure or know what are the different parts of the pH electrodes. Okay, electrode head, and this is your filler opening, and you can also note down what is the reference electrode and why the internal buffer inside, and internal reference electrodes, and what is the separator, and also the glass membrane. Okay, so these are typical. The parts are contents in the combined electrodes. Okay, yeah. So we'll move on now. The determination of pH, that you need a pH meter, standard flask, magnetic stirrer, funnel, beaker, wash bottle, tissue paper, and also forceps. So these are the materials required or apparatus required for their pH measurement. Okay, pH meter is for measurement, and standard flask for is making your standard pH solutions. Magnetic stirrer for complete mixing, and then tissue paper for wiping out your electrodes, and forceps this is for picking up the pH buffer tablets. Okay, yeah. These are the chemicals required. Number 1, please make a note on. For the standardization of your pH meter, you need to have 3 different pH range. One is in the acidic range, second one is about the neutral, and third one is about your basic condition. So 4, 7, 9.2. Okay, some books they may refer 4.01 or 4.02. Second one is 7 pH, and third one is about 9.2. And second one is about potassium chloride, and third one is about distilled water. Okay, so please remember, you can also get the pH, the standard solutions in bottles like this, or you can also have tablets like this, pH tablets. Okay, so you could be able to identify or differentiate by colors. 4 by orange color, 7 by green color, and 9.2 is by blue color. Is it okay, Sharon?
Yes, sir.
Yeah, 4, 7, and 9.2. Please remember, you can also take directly from a solution, which is all commercially available, or you can prepare from tablets 479.2. Okay, these are the three chemicals are required. Okay, so now we'll move to a sampling protocol and also preservation. Okay, yeah, preservation of sample is not a practical. Okay, so you need not preserve the sample and go for a pH test. Immediately after the sampling, you can go for a pH measurement. Please remember why preservation is not practical as the imagine now if you are taking a sample in a contaminated area, there could be a biological activity or microbial activity, and there could be a changes in quality. Please make a note on biological activity will continue after the sample or sampling. Changes may occur during handling and also storage. Okay, that is also one fact. And to reduce the change in samples taken for the determination of pH and keep or preserve the samples at 4 degrees Celsius. Please make a note on. For larger sample, preservation is not at all practical, but assume that it is also been contaminated with some sort of biological activity. What do you do? Immediately after the sampling, you keep it in a freezer, okay, with a temperature range of 4 degrees Celsius. So in the 4 degrees Celsius, microbial populations are highly not reactive. Okay, it will be there in the system, but it may not be very reactive. Thereby, it may not induce any changes in the quality. So please make a note on 4 degrees Celsius. Most of the water sampling or preservations are at 4 degrees Celsius, 4 degrees Celsius, and do not allow the sample to freeze. That is also another important fact. And most of the cases, it is being recommended to analyze the sample as soon as possible. Okay, so these are the sampling. Okay, so you just go with the sampling, say example device. Okay, and sample in a clean container and take it to the laboratory and do the test immediately. And if it is not favorable conditions, then you can also preserve the sample at 4 degrees Celsius, which will arrest your microbial activity. Yeah. See, these are the precautions. Please make a note on. Number one is temperature. Okay, temperature plays a major role. Temperature plays a major role in the measurement of pH. Okay, so please make a note on. Number one, the first is caused by the change in electrode output. Please make a note on electrode output at different temperature. Okay, probably you would have brought the sample at, say example, 27 degrees Celsius. In the laboratory, maybe 24 degrees Celsius, and there could be a changes in pH value what you are expected. So please make a note on the temperature changes plays a major role in the electrode output. That is number one. That's why during measurement you should also monitor the pH of the temperature of the sample. Okay, so number one. Number two, the pH inherent in the sample at different temperature. Okay, so that is also another fact. So please make a note on. Thereby, the pH as well as temperature should be noted during the analysis of your pH or during the determination of the pH value. So please make a note on. Number one, pH will give you, temperature will alter your electrode output, and also it is also being greatly influenced by your pH of the sample. That's what now during measurement you should also note down the temperature of the water. Okay, yeah. The second point, number one, temperature we have mentioned, temperature. Number two is the glass electrode, because you are doing in a glass electrode, and the solution should not contain any amount of color, salinity, colloidal matter, oxidants, turbidity or reductants. Please make a note on. Okay, this is another criteria. Okay, so water should not contain any amount of color or turbidity or high salinity or high colloidal matter. And anyway, you are going to measure with the glass electrode, and the water should not contain oil as well as grease. Otherwise, it will form a thin layer on the like your combined electrode, which may also sometimes will give a erroneous results. And before before your pH analysis, and oil and grease should be removed. Okay, or after measurements, you have to wipe out completely with detergents. Please make a note on. The electrode should be wiped off immediately after measurements. Okay, and the before measurement, the fourth the precaution is before measurement, and the electrode should you should keep the electrode at least 2 hours in a dilute hydrochloric acid. That is also another one criteria. and electrodes used in the pH meter are highly fragile. Hence, the handle it very carefully. So these are the precautions or some important points you have to note down. Number one, temperature. Number two should not contain any amount of other agents like color, salinity, colloidal matter, oxidants, turbidity, and reductants. It should also have any amount of oil as well as grease. Okay, yeah. So this is a procedure. Three different procedures. Number one is preparation of reagent and calibration of your instrument. And third one is about your testing. Okay, so please make a note on. So now you have to prepare a buffer solution of pH 4. So what you have to do now? You have to take out 4 buffer tablets and take 100 ml of standard measuring flask and place a funnel over it. You can drop and you can use as forceps. Take a pH tablet of 4 and allow little amount of water in the okay, let me draw a diagram. This is your measuring jar and this is your funnel. Okay, so what do you do now? You just place the buffer tablet here and pour water, little amount of water, and make up to 100 ml. Okay, this is what you have to prepare a pH of 4 solutions. Very simple. Okay, you just take a conic measuring flask of 100 ml and place a funnel over it. Use a forceps and take a 4 buffer tablet, drop it here and pour water. Automatically this pH tablet will dissolve, and add further water and mix it carefully and make up to 100 ml. So this solution corresponds to 4.01 or 4.1 or 4 pH value. Is it okay? Is it fine, Sharon?
Yes, sir.
Okay, so number one. Number two is now I have a buffer of 7 as well as 9.2. Now you have to repeat the same procedure. Okay, buffer number 7. What do you do? Now you take a buffer of 7, pH buffer tablet of 7, and then prepare 100 ml of sample with the 7 buffer tablet, and this will corresponds to your pH of 7. If you take a sample from this prepared like solution, if you measure a pH, it will sometimes say 7 as your pH. Third one, 9.2. Okay, so what do you do? They repeat the same procedure. Only the tablet is now 9.2. Okay, and make up to 100 ml, and that will represent the pH value of 9.2. Is it okay? Let me repeat again. Anyway, you'll also like see some videos. Okay, so what you do is take a measuring flask of 100 ml, place the funnel over it, use a forceps, take a buffer tablet, drop it in the funnel, you add little amount of water, dissolve the buffer tablet, okay, then add further distilled water and make up to 100. So this solution corresponds to 4. Repeat the same procedure for 7 as well as 9.2. So now you have a 3 different set of solutions with pH of 4, pH of 7 and pH of 9.2 is ready for calibration. Is it okay?
Yes, sir.
Yeah. So now, before going for the measurement, you should also calibrate the instrument. Calibration is the important part. So what is this calibration? It's very important. Calibration is very easy. I have a standard solution of 9.27 and 4. And if I place this pH solutions in the electrode, it should also read out the same value. If it is not reading out as same value of instead of 9.2, it is showing sometimes 9.3 or 9.1. Okay, you adjust the calibration of, you make it 9.2, 4 as well as 7.2 for 3 different calibration sequence. Once the equipment is calibrated, you can go for a direct measurement. So let me repeat, or let me go for a calibrating the instrument in step number 1. So what you do now, in a 100 ml beaker, what I have prepared, take a BH 9.2 buffer solution and place it in a magnetic stirrer and insert the Teflon coated stirring bar and stir it well. Please make a note on this. You have to take it in a beaker. Okay, so 9.2 it is in a conical, it is in a measuring flask, and take it a smaller quantity inside, and you place a Teflon coated stirring bar and place it over in a magnetic stirrer. Switch on the magnetic stirrer, automatically it will give a complete stirring. That means you need to have a homogeneous mixture of this sample, Then you place your electrode and read it. That's all. Okay. So place the electrode in the beaker containing the stirred buffer and check the reading in the pH meter. And if the pH meter is not showing the value of 9.2, what do you do? Now you adjust the calibration knob. That is something important. Please make a note on. You just adjust the calibration knob, adjust to 9.2. Then take an electrode out and wash with water. That's something is important because this electrode outside is being mixed with 9.2 of buffer solution. Probably next calibration you may go for 4 or 7. That normally the solution which is on the surface of the, say example, electrode may also hinder your results. So please make a note on. Once after the calibration with 9.2, take out the electrode out and wash it with distilled water and wipe gently with a soft tissue paper. That's what we have seen in the equipments or apparatus required. Repeat the same procedure for again 7 buffer tablets as well as 4 buffer tablet. Is it okay? So what you do now, step 2, and take a 7 pH buffer solution and take it in a beaker, and even inside you make you just add your Teflon coated stirring bar. Keep it in the magnetic stirrer, switch on the magnetic stirrer, automatically it will go for a stirring process. Place the electrode and read the pH value. If it is a 7, you need not worry about. If it is a 7.1, you adjust into a 7 value. Thereby the instrument is again calibrated with 7 pH value. Step number 3. Okay. Step number 3. Take a pH solution of 4 and go for a complete stirring and read the pH value. If it is 4, is fine. If it is 4.1 or 3.9, you adjust the calibration knob and then you make your calibration correct. Now calibration is completely being done with pH of 4, pH of 7 as well as pH of 9.2. Now your equipment is ready for your measurements. Okay. Now what you do? Take out the sample. Repeat the same procedure again. Take out the sample. Please make a note down. Here is testing of the sample. That means is what you are going to measure the pH of the sample. Maybe with the water or maybe with the wastewater or maybe with the acid solution. So here also you have to do the same procedure. What you do now? Instrument is already been calibrated. Take a 100 ml in the beaker. 100 ml in the beaker and add your Teflon coated stirring bar. Switch on the magnetic stirrer. Automatically it will mix your sample and keep your calibrated the pH electrode inside. Okay. Then you just wait for the stable reading. Because of the mixing, there could be alterations or changes in the pH value, and wait until you make it a stable reading and note down the pH of the particular sample and take the electrode out after the measurements and wash it again with distilled water. You may not know what type of the sample it is, and wipe it gently with a soft tissue and keep it in a electrode stand. Okay, this is what normally they do it in a laboratory. Okay, yes. Then what you do now, the calculation that I already mentioned to you, you have to also measure or note down the temperature of a particular sample. Okay, sample number 1, 23 degrees Celsius, and pH of the sample, say example 6.9. Okay, so you can note down these values for your tabulation. Okay, yeah. So this will give a comprehensive idea of the procedure. Okay, so what's what I told you. Switch on the instrument pH meter at least 30 minutes before your test and prepare the standard buffer solution of 4, 7 and 9.2. Okay, 4, 7 and 9.2, and you can also calibrate the instrument with 9.2, 7 as well as 4. Please make a note on what I mentioned is 9 you prepare, keep it in the sample like this. Okay, this is what I mean to say. It's a magnetic stirrer. This is your electrode. Okay, so magnetic stirrer is for mixing, electrode for the measurement. If it is, say example, if it is 9.3, so you adjust this calibration knob and make it 9.2. Take out this electrode, wipe it, and place it into a next buffer solution of 7. And read if it is 6.9, you adjust, 7.1, adjust. Otherwise, if it is 7, you need not do anything. And third buffer, calibrate. And so once the calibration is being done here, you can go for a measurement of the sample. Thereby, you can read out the pH of the solution as a particular value, sample as 8.2. Is it okay?
Yes, sir.
Yeah. So please make a note on. So each and every experiment I will also project like this as a procedure chart. You can remember very easily. So chart, prepare a solution, buffer solution with 4, 7, and 9.2. This will be available in solution, or you have to prepare through buffer tablets. And you can now you have to calibrate the instruments with the 3 different buffer ranges of 9.2, 7, and 4. Once the instrument is calibrated, now the instrument is ready for n number of measurements. N number of measurements. Okay. Yeah. These are the different pH ranges. Please make a note on this. I already mentioned to you. Gastric acid to the bleach, and what are the food, what do you eat? You should also have some idea on what's the pH ranges. Okay, so most of the food what we eat in this range of this. Okay, 3 to 10. Please make a note on those who have addictions towards soft drinks, and your pH range is always in the range of 3 to 4. And if you happen to eat a lot of fruits, it is almost in the range of 8 to 9. And if you happen to eat a lot of meat and other non-veg item, it is in the range of 5 to 6. So please make a note on this pH ranges. Okay, this is the typical pH meter. Okay, this is what I told you. Number one. Okay, this is the digital pH meter. Nowadays, you are getting with the different advanced instruments like the pH meters like this also. Okay, this is what I told you is about pH combined electrode. This you have to keep it in a solution all the time. Okay, and this you can see now the calibration knob here, and this is your display unit, and if the based on your buffer tablets, you can also calibrate the instrument for 4, 7, and also 9.2. Okay, so nowadays this is very simple, and you may get a lot of handheld pH meters also. Okay, it's not very big. Probably in the laboratories you could find, but in the household level, people used to have handheld pH meters like this. Okay, these are almost 2,000 to 3,000 rupees. You can also carry something like a pen. Okay, you can also measure pH of the sample at any point of time. Okay, this is what large amount of handheld pH meters are available for pH measurement. Okay, so this covers your pH experiment. pH experiment. Yeah. Let me share a small video now. Yeah. Is my screen visible, Shravan?
Yes, sir.
Shravan?
So your screen is visible, sir.
Yeah. At least you can listen for 5 minutes.
Sir, but the audio is not.
Audio is not there. Sharon, is it okay? So you could able to now familiar with the pH of the sample or pH determination. Okay, so Dr. Yukesh will share the individual pH values because it's nothing very serious, but at least you should also understand logically how the pH is being done in the laboratory. Okay, so we'll share for individual roll numbers with the individual pH value, and you can refer the standard you need not copy, but you can also showcase your originality in writing records also. Okay, it should be unique in nature. And based on that, we can submit as the experiment number one. Experiment number one, determination of pH. So any clarifications here? Yukesh, you wanted to say something?
I think, sir.
Yeah. Yeah. Sharon, is it all right?
Yes, sir.
Yeah. So at least you can make a two set of two sheets, A4 sheets. You can write aim, apparatus, and principle, and the procedure. And the main important point is about your observation, nominal significance, and at least by seeing the value, you could also say something about the nature of the sample. That is more important for the examination point of view, or your uniqueness, how you have understood the problem statement very nicely. Is it okay?
Okay, sir.
Yeah. Move on to the experiment number 2, determination of turbidity. Okay, second experiment, determination of turbidity. Okay, yeah. Okay. Okay, so those who are familiar in physicochemical process or water treatment, and turbidity, last semester you would have studied, is the technical term referring to the cloudiness of a solution, and which is also imparted by a solid particle, and which may also obstruct the transmittance of light through a water sample. Please make a note on cloudiness or haziness, that probably would have studied in the last semester. Cloudiness of a solution, and which is imparted by a solid materials or solid particles, and which sometimes obstruct the transmittance of light through a water sample. Okay, so main turbidity causing agents in the water are clay particle, organic matter, silt, algae, and other microorganisms. If the water contains these substances, you could expect turbidity in the water. Okay, clay and silt is very common because of surface runoff. Organic matter also again from surface runoff into water bodies, and algae probably with organic contamination or by nature in the water bodies, and other microorganisms, which causes turbidity of water, turbidity in the water system. Okay, yeah. Why turbidity is important? Because the turbidity causes the aesthetic displeasing appearance. Please make a note on. Okay, if I have a glass of water and certifies that water is good in all quality, but we have some sort of, say example, turbidity in sense. Okay, turbidity water, and probably people may not take it because of aesthetically displeasing appearance. Please make a note on number one. Okay, because turbidity may not have a problem, but it will also create a situation aesthetically displeasing opaqueness or milky coloration in apparent. Okay, so turbidity normally create a lot of this problems, aesthetically displeasing opaqueness. Okay, and the colloidal material which exerts turbidity provides adsorption sites for chemical and biological organisms. Imagine our situation: if the water contains lot of organic matter, it's a solid. So what will happen? These microorganisms or the bacteria which is surviving in the water, which will attach onto a surface of the organic matter. Okay, it will also play something like called hide and seek. Okay, so say example, if you purge say example UV light for the disinfection of microorganisms in this direction, what will happen? This organic solids will act as a protective layer which may not allow the UV light to kill all these microorganisms. That's what something is important. Please make a note on. These are colloidal material which exerts turbidity, provides adsorption sites for chemical and biological organism that may not be harmful. They may not be harmful. Okay, and this These turbidity causing agents will also cause undesirable taste and also odor. Imagine now if the water contains a lot of organic matter, there could be a lot of undesirable smell and also taste. You would have come across in water bottles. Water bottle, if you have surface water, you take a surface water in a lake or something or a good water and keep it ideally for 2-3 days. If you open up, you may get a smell. Okay, because of these conditions, undesirable taste and also odor. Okay, and please make a note on this. What I told you, these solids may partially shield organisms from a disinfectant. That's something is important. This point is very important. You may feel like I have done a disinfectant nicely, but once you consume, there could be a possibilities of infections. Okay, these organic solids will act as a shield. Thereby, these organisms will escape from disinfection activity by either by chlorination or by UV disinfection or ozonation or whatever may be. Please make a note on this point is very important. Okay, and natural water bodies, turbidity may impart sometimes is brown color or other color to water. Okay, main problem with the turbidity is only color. You may get a brown, say brown color water or yellow color tint water, or sometimes it's milky white. That is because of these turbidity causing agents. And the main important problem of this turbidity in main huge water bodies are it will not allow the light to penetrate inside. That something is important. Light penetration will be hindered by these particles. Thereby, the photosynthetic action by this algae and other organisms will deprive of. That something is important. Please make a note on. These turbidity causing agents will not allow the light to penetrate inside the water bodies, and thereby the photosynthetic reactions in streams and also lakes by organisms is greatly affected. That is number one. This is another one important feature with respect to turbidity. Okay, and imagine our situation like the water contains lot of turbidity. Okay, and it is unnecessarily increase your load on slow sand filters. Okay, probably would have come across in this last semester. If you want to polish the water, and the final ultimate treatment option is filtration. You just allow the water to pass through a filter, and because of some mechanisms, and these particles are getting trapped onto a surface. Okay, imagine our situation. The water is coming with lot of turbidity, and thereby it will also increase the unnecessary loads in the slow sand filters. Okay, yeah. Thereby, the knowledge of the turbidity requires special treatment by coagulation and also filtration. Okay, that another important point. So turbidity plays a major role for defining your treatment efficiency. Say example, chemical coagulation and also filtration. And turbidity also sometimes is used to determine the effectiveness of treatment produced. Say example, if you have a water treatment chain like coagulation and filtration and disinfection, and your treated water, and you can measure the turbidity here. You can measure the turbidity this point. You can also measure turbidity this point. This minus this will give a treatment efficiency, and you can also monitor the turbidity of water at the end of the treatment. If it is any increase in turbidity, you can also look back immediately. The filter is not working properly, or whatever you are adding coagulant in the water is not sufficient. Okay, so please make a note down. This concept is very important. Okay, to determine the effectiveness of treatment produced with different chemicals and dosages needed. And turbidity measurement help to gauge the amount of chemical needed from day-to-day operation in water treatment plants. So very easy. If you want to check the capacity of the plant, the easy way is to measure turbidity. That's what I told you. This story: coagulation, filtration, disinfection. Take out the sample in the treated water, measure the turbidity. If it is anything on turbidity changes, you can immediately look into the treatment option like filter, whether the filter is running properly, or whatever the coagulant you are adding into the coagulation chamber is right or wrong. You can do it very immediate. Okay, yeah. And the measurement of turbidity in settled water prior to filtration is useful in controlling chemical doses also. That's what I told you. And turbidity measurement of the filtered water are needed to check the faulty filter operation. That something is important. Okay, if you take out the sample here, if it is represents with lot of turbid or turbidity, then you can look back very immediately with the filter operations. There could be a damages in the filter, or the filter is not working properly. Okay, yeah. And you can also measure optimum coagulant dosage Okay, optimum coagulant dosage by turbidity measurement. That's also you would have seen in the last semester. And optimum coagulant dosage. So what do you do now? You can add coagulants into the water. You can measure the remaining turbidity. You can also measure turbidity efficiency. You can also measure like this. So this could be your optimum coagulant dosage. This and all you would have studied in the last semester. Okay. So optimum coagulant dosage is measured in particular with the turbidity remaining in the water, or what is the efficiency of the turbidity removal. Okay, please make a note on turbidity measurement is used to determine the optimum coagulant dosage of the particular water. Okay, for the particular water or wastewater, and you can also measure or evaluate the performance of water treatment plants by measuring simple turbidity. Value. Okay, so these are some of the anomalous significance of turbidity value. Okay, yeah. Here also again the principle, and principle is probably you would have come across the measurement of turbidity by photometry and nephelometry. Okay, so these two concepts you would have studied in the last semester: photometry and nephelometry. Okay, nephelometry. So photometry, just you have a sample and you just pass the light, and you can measure the absorbance of light just opposite to the direction of light. That is called photometry. That you would have studied in the last semester. And if you measure the scattering of light or its scattered intensity perpendicular to the direction of light, that is called nephelometry. That we are not going to discuss in detail, but at least you measure the basic principles of turbidity measurement is either by photometry or nephelometry. Photometry is direct, just opposite to your light source. You can measure the scattered light intensity, and if you measure the scattered light intensity perpendicular to the direction of light, that is called nephelometry. So please make a note. That's what the first point says. Turbidity is based on the comparison of the intensity of light scattered by the sample under defined conditions with the intensity of light scattered by the standard reference suspension under same conditions. That will come back later. What is the sample and also what is the reference standard? Okay. The turbidity of the sample is thus measured from the amount of light scattered by the sample, taking a reference with the standard turbidity suspension. So probably you would have come across the different units associated with the turbidity are turbidity unit and Jackson turbidity unit and Formazin turbidity unit and Neflo turbidity unit. So these measurements you would have come across in the last semester. Okay. So turbidity unit is very simple. If you take 1 milligram of SiO2, dissolve in 1 liter of water, it may represent some turbidity that you can use or measure in terms of 1 turbidity unit. So please make a note of this. Something is important. Standard turbidity, 1 turbidity unit is 1 milligram of SiO2 in 1 liter of water, which may represent some turbidity in water. That is called turbidity unit, 1 turbidity unit. Okay, you would have come across Jackson also. Anyway, this we are not going to discuss. Jackson turbidity unit. And if you use silicon SiO2, you may not get reproducible results. And for that, people may use Formazin. Okay, people may use Formazin. If you are using Formazin as a standard solution, that is called Formazin turbidity unit. And Neflo turbidity unit is by nephelometry. So these are the basic concepts. You can also look back what you are studying in the last semester. Okay. So the third point, the higher the intensity of scattered light, the higher is the turbidity. And you can also use Formazin. That's what I told you. The problem with silicon dioxide is you may not get a reproducible result. Okay, if you want to get a reproducible result for a same sample, and you can use Formazin instead of silicon dioxide, that is called Formazin turbidity unit. Formazin turbidity unit. Okay, yeah. So these are the materials or apparatus required. You need to have a turbidity meter, standard flask, funnel, a wash bottle, and also tissue papers, similar to your pH measurements. Okay, these are the chemicals are required. Number 1, hexamethylene tetramine, hydrogen sulfate, and also distilled water. So you can also the chemicals also available in standard packs: hexamethylene tetramine and hydrogen sulfate. Okay, so by using these two chemicals, now you have to prepare a standard. Okay. So here also again sample handling and also preservation is almost similar to the pH experiments what we have discussed. And water sample should be collected in a plastic can or glass bottle. And all the bottles must be cleaned thoroughly and should be rinsed with the turbidity free water. And volume collected should be sufficient to ensure a representative sample. Allow for replicate analysis. At least you need to have a two testing. Okay, duplicate testing. And there is no chemical preservation is required. And if you want to store the sample, you can keep it the sample at 4 degrees Celsius. Don't allow the sample to freeze. And analysis should be carried out immediately after the collection. And if you want to keep the sample, or if you are going for a turbidity measurement after a day, better to keep the sample at 4 degrees Celsius, and you can keep it maximum of 2 days, 48 hours. So these are some of the sample handling and preservation requirements. Okay, yeah. And these are the precautions. Number one, the presence of colored solutes causes measured turbidity values as very, very low. Okay, that's what something is important. You should be cautious with the color in the solution or the sample. And precipitation of dissolved constituencies sometimes also cause your turbidity in a higher value that may give erroneous results. Light absorbing materials such as activated carbon in significant concentration can cause very, very low readings. And presence of floating debris and coarse sediments which will settle there, that also reflect again your value as very, very low. And finally, divided air bubbles can cause higher readings. So these are the precautions you should note before going for your measurements. Please make a note on. You should observe the sample as is colored or not. Is there any precipitation happening? And is there any light absorbing chemicals? Please make a note on. Thus, photometry or nephelometry is by the principle of scattering of light. Okay, if you should also find out if any light absorbing materials are available in the water or not. Okay, that is the third point. And fourth one, water should not contain any amount of floating debris, and which may settle inside your sample bottle. Obviously, you may get a lower results or lower reading. And last one, finally, divided air bubbles can cause higher readings. So these are the precautions before going for your measurements. Okay, now the procedure, you have to prepare the reagent. Number one, you have to prepare a hydrogen sulfate solution. You take 1 gram of hydrogen sulfate and add 100 ml of distilled water, or take 1 gram of hydrogen sulfate and precisely weigh it and transfer into a 100 ml standard measuring flask and place a funnel over it and add with distilled water and make up to 100 ml. So this is the preparation of reagents number one, hydrogen sulfate. And second one is the hexamethylene tetramine. Here you have to take 10 grams of hexamethylene tetramine and take 100 ml of standard measuring flask and place a funnel over it. And then you add, dissolve this 10 grams of hexamethylene tetramine and make up to 100 ml in the standard measuring flask. That's something important. This is what something similar to the pH what you have. So this is your okay, standard measuring flask with 100 ml as mark. Okay, then you can take 1 gram of hydrogen sulfate. Then this may be the powder. Then you add distilled water, and this salt will go. Suppose this salt will dissolve and add further into water, and you make up to 100 ml. Okay, this is solution number one. And here again, take a round flask, round bottom flask, and place the funnel over it, and take 10 grams of hexamethylene tetramine. Add water and dissolve this 10 ml completely, 10 gram completely, and make up to 100 ml. So this is solution number two. So what do you do now? You just mix 5 ml of solution number one and 5 ml of solution number two. Okay, so what do you do now? 5 ml plus 5 ml of solution number two, and this is number one. Okay, and take it into a round flask of 100 ml. Okay, this contains this 10 ml now, and add with water and make up to 100 ml. Okay, and allow this mixture of 100 ml for 24 hours. 24 hours, and if you take out the sample at after 24 hours, and which will give a standard solution of 4000 NTU. Please make a note on that. Something is important. Is it okay, Sharon?
Yes, sir.
Yeah. So 5 ml of this is solution number 1, 5 ml of solution number 2, and allow the mixture for 24 hours. There could be a turbidity production. Okay, and you make a final volume of 100 ml. Okay, with turbidity free distilled water, and that standard solution will give you 4000 NTU, 4000 NAFLO turbidity unit, NAFLO turbidity unit, 4000 NTU, and with that 4000 NTU, I could able to dilute, and I may also have a different ranges of 440, 400 or 4000, whatever may be. In any combination, if I want, I can prepare by simple dilution methods. Okay. Yes. Now the standard solution is ready. Now you have to go for a standard calibration. So what do you do? This is your this is your turbidity meter. This is called cuvettes. Please make a note on. This is called sampling bottles. This is called a cuvette. Okay. You could also see now two different knobs here. One knob is for your range. Okay. Please make a note on one for your range. I told you, 440, 400 and 4000. So in which range you are in a position to say approximate range of the sample. Thereby I can able to fix this range. Number 1. Number 2 is this is for your calibration knob. And this is for this is again two knobs here to set 0. Okay. So what I do in the turbidity measurements, and you have to calibrate the instrument by standard NTU as well as distilled water. This will give 0 value. Distilled water, this turbidity free water should show 0 value, and this standard NTU you have to adjust with calibration knob. Is it okay? So you could able to see 4 knobs. One is for range, others is for calibration, and this is for to set 0. One is for maximum adjustment, that we call it as coarse, and this is for fine measurements, fine adjustment. Okay, if you want to adjust coarse way, then you can use this knob, and if you want to adjust very fine, you can use this knob. Okay, these are standard cuvette bottles. Please make a note on. These are cuvette bottles, and you will be able to see the mark. So what you do now, you have to fill the sample only up to this mark. And you could also be able to see now a black color lid. Okay, you can remove and place this bottle inside. Okay, and you can also like close the sample with this black box again. That means you should not allow the external light to hinder your activity. That's something is important. Please make a note on. This is by either photometry or nephelometry, and external light should not disturb your turbidity measurements. Please make a note on. This is what I told you. 2 knobs in the bottom to set 0, and 2 knobs at the top. One is for your range selection, others is for your calibration knob. Okay, so for this problem, I adjusted to, say example, 1000 NTU. Okay, so I have taken 1000 as the particular range. 1000 as a particular range. Okay, so now what do you do now? Now what you do? You just take a turbidity-free distilled water. Turbidity-free distilled water up to the horizontal mark. Place the cuvettes in the turbidity meter. So that the vertical mark in the sample cell should coincide with the mark in the turbidity meter. Please make a note on. Let me draw this. That something is important. Okay, so let me draw the top opening of the cuvettes. Okay, say example here. Okay, and one mark also will be placed here. You could also see one mark here also. Okay. So in the bottle also there is one horizontal mark, and also you could able to see one vertical mark also. So what you do now, you place the bottle inside. Okay, and this mark will coincide with this directions. Thereby, whatever the principle what we have discussed, the transmittation of light, and thereby you can measure the scattering of light exactly perpendicular to the direction of light. That something is important. Please make a note on. So these two are important. One is this horizontal mark for your maximum sample limit. One vertical, this vertical, and the horizontal mark which is placed inside in the, say example, the equipment should coincide each other. Okay. So please make a note on. So now what I what I have taken is only distilled water, which is free from suspended solids or free from turbidity. And now note down here, you just observe the value. You should read only zero, only zero. If it is not zero, you adjust this either of these knobs to make it or read it zero value. Please make a note on this is number one. Number one, with distilled water, you should read zero. If it is one or two or three, you adjust this knobs to set zero value to zero value. That something is important. Okay. Now, once after that, you take the 200 NTU. That's what I told you, because I have already fixed 1000 as range. I have prepared 200 NTU from 4000 NTU of standard solution. Okay. Now I'm going to calibrate the instrument with 200 NTU solution. Okay. So now what I do, I'll take 200 NTU in the sampling bottle with this mark and place it inside the turbidity meter. Close the lid. And what I do now, I may adjust this calibration knob to read 200 in this reading value. That's something is important. Please make a note down. If I want to make zero, I can use this two knobs. If I want to calibrate the instrument, I may use this calibration knob to set 200, because I have a standard solution of 200 NTU. If it is 20, you have to read with 20. If it is 2000, you have to read with 2000. So depending upon your sample turbidity value, you should also in a position to fix the standard solution. Okay, yes. Now repeat the procedure for 2 or 3 times. So what you do now, you just take out the sample, the standard NTU solution and keep it again one more time with distilled water, and you just observe whether it is reading zero or not. If it is not zero, adjust the bottom knob and make it zero. Replace the vial and place again 200 one more time and do the process zero again, your standard 200 NTU for 2 or 3 times. And if it is correct, then you can fix the instrument is calibrated. Once the instrument is calibrated, now you can go for a testing of sample. So you have unknown sample with you now, and take a sample in the cuvette and up to the mark and place it inside the turbidity meter and wait until you may get a stable value and note down the turbidity of the sample with the turbidity meter. Okay, so these are the so please make a note on calibration of the instrument is very much important. One is this is preparation, calibration, and your testing of your water sample. Okay, the standard solution is here 200 NTU, and your normal zero turbidity solution is distilled water. Calibrate the instrument with 0 NTU as well as 200 NTU. But repeat the procedure for 2-3 times. Once the instrument is calibrated, you can go for n number of measurements with this turbidity meter. Okay, yeah. So this is again the procedure chart. Switch on the instrument, and you may take any standard solutions. Here in the chart, I mentioned 400 NTU, and you just calibrate the instrument with 400 NTU, and calibrate the instrument with 0 NTU with the distilled water. Repeat this process for 2-3 times. Once it is calibrated, then you can go for a sample turbidity measurement without any difficulty. Is it okay?
Yes, sir.
Yeah. So here also again, you can also measure or note down the temperature of the sample, and you can also measure the turbidity in terms of nerve flow turbidity unit. Okay. So these are the typical turbidity meters are available in the laboratories. Okay. This is what I told you, the cuvette bottle. This is your turbidity meter, and turbidity meters also now this is available in handheld also. It's very easy to carry and very small instruments. Okay. So these are the cuvettes. And you may get a standard solution and also distilled water and place it and make it 0 and make it your standard NTU value once it is calibrated, and you can take out the sample. You can measure for the turbidity for n number of measurements. Is it okay?
Yes, sir.
Yeah. So this will cover your the turbidity. So let me play one 3 minutes video. Yeah, you can look on. Yeah. Okay, now is it all right? So whatever we are seeing is two different experiments. One is this determination of pH and determination of turbidity. Is it okay?
Yes.
Yeah. Vignesh? Vignesh?
Yes.
Yeah. Sudarshan?
Yes, sir.
Yeah. So now Dinesh?
Yes, sir.
Yeah. So this is now so A section, Sharan and Vignesh will follow, and Sudarshan and Dinesh for B section. So now the Dropbox is ready, and now whatever we have discussed so far, this is pH experiment and the turbidity. So individual values will be shared shortly with the individual pH value and also the turbidity value, and you can start preparing on your record note. Okay, at least for 2 A4 sheets, and please write very neatly because it carries 40%. And take A4 sheet and have a border and write. Okay, whatever I have given in the, please note, whatever I have given in the manual is by instructions. Okay, please take note down or rinse. Okay, now you have to write in a passive voice because you have completed the experiment. So please make a note down, don't copy it, and make a passive voice, make a note, passive voice way of writing. Okay, so sample is taken, the reading is being noted. Okay, and the instrument is calibrated. Something like you just change the words and write it. Understood that you have done the experiment. Is it okay?
Yes, sir.
Yes,
It's
sir.
fine. Yeah. Any clarifications here? You can post it now.
So we
Thank
have
you.
to only
Yeah.
write the experiment.